We live in the information age, and a consequence of this is that we've become rather dependent on search engines like Google for any sort of doubt we have about our memories. For example, have you recently tried to remember the name of the actor starring in the movie you saw and just said, well, I'll just Google it? Because before the dawn of smartphones, we relied on our brains to do most of the documenting of our life and to store information. And the brain is wired to collect information about our lives and store it as memories. Memories allow us to have a perspective on what might happen to us based on our previous experience, and in this way, memories influence our choices and behaviour. So different areas of the brains have different functions, and one area we know which is important for learning and memory is called the hippocampus. So like a web page, information can first be acquired by the brain and then encoded. And the hippocampus acts to do this because it's like a hub for the brain. It's the Google which connects lots of different areas to each other like hyperlinks. So to actually connect two brain regions, this happens at the level of the brain cells, which are called neurons. They connect to each other at junctions called synapses, and a synapse can either be strengthened or also weakened, and this is called synaptic plasticity. In this way, the brain can change its connections in response to our experiences. For example, the signals that neurons receive are called neurotransmitters, and an important one is when we receive a pleasant surprise, like a bar of chocolate, we receive a wave of a neurotransmitter called dopamine. Now, dopamine can modify the neuron by binding to these tiny protein structures on the surface of the neuron called receptors. Receptors catch on to the neurotransmitter like keys fitting into a lock, and once they activate the receptor, they trigger a cascade of signaling within the cell that itself can activate genes and lead to the production of new proteins. Now, in the laboratory, we have certain drugs available to us which can prevent this production of proteins called protein synthesis inhibitors. And interestingly, if an animal has been trained in a spatial maze and then is administered protein synthesis inhibitors, the next day the memory of the maze will be gone. This showed us that the consolidation or the storage of the memory is dependent on protein synthesis. But what about updating our memories? Because at the same time, we can't retain all the information that comes into our lives. That would be just too much information. Like the tweets of Miley Cyrus, we don't need to know everything that happens to us. And one way to do this is to weaken the strength of connections, to weaken synaptic plasticity. And this is essentially forgetting. We think that this happens because there's a deficit in reactivating our memory because the connections have been lost. On the other hand, you might want to update your memories and include new information that's relevant to your past. Importantly, the reactivated memory, the neuronal network that becomes reactivated in the brain, can not only lose connections, but can also form new ones and integrate new information. So a reactivated memory is really reconstructed from whatever elements of the story are left connected. And importantly, we know now that there are distinct cellular mechanisms responsible for the original consolidation of memory, so the original storage when you learn something new, and its reconsolidation, or the restoring of the memory after it's been reactivated and new information has been laid down. So this leaves open a new therapeutic goal for certain psychiatric disorders where we think memories might be hyperactive or we would like to actually reduce them. For example, a very strong memory that is easily learned and retained over life is fear. And a fear or stressful experience releases adrenaline. Adrenaline is also a neurotransmitter, and it's a close cousin to dopamine. It has its own set of receptors that it binds to, and in a similar way, it can modulate synaptic plasticity. Importantly, we can train animals in the laboratory to be what we call fear conditioned, or to associate a fearful experience with a certain cue. A commonly used paradigm is that we place the animal into a cage that has a wire grid floor, and we play, for example, an acoustic tone. Right after the tone, a small electric shock is given to the animal, and the next day, to test how well it learned the association between the tone and the shock, we replay the tone again, And even if there is no electric shock, the animals present a fearful behaviour which we call freezing. This is what rodents do in response to a predator. They stop moving. And this is a measure of how well they've actually learned the association between the tone and the shock. Importantly, we can use this paradigm to investigate whether we can reduce fear memories by using drugs that specifically target the receptors for neurotransmitters like dopamine and adrenaline. And in this way, we might even be able to modulate and modify memories that have already been formed, because when they are reactivated, as I mentioned, they become open again, so they can either be weakened or strengthened. If we target memories at this stage, we'll be able to essentially hack memories and alter what our brain holds on to in the future. And this is a real therapeutic goal for certain disorders like post-traumatic stress, where we think the fear memory is too strong and it becomes generalized and overactivated. So in this way, we're not too far from understanding better how we might eventually hack our memories.